Hey guys, welcome back to my channel. So till now what we did is we looking for the datasets which is related to the Parkinson. We explored the GEO gene expression omnibus and then we select the Parkinson database and then run this database with GEO2R. When GEO2R from we download the table of the gene expressions, totally differential expressions genes, then we copy into the Google spreadsheet. After that, once we spreadsheet, we manipulate the data according to the 0.05, and then we playing with the data with the string database where we can find the different pathways and how we can export the clusters. And then on the basis of that, we also use the Panther database by navigating through the gene ontology, and we put some genes over there and then powered by Panther, you can see. And then we can biological process, then we launch the process and it will give us the windows where you can find the GEO gene ontology data, and you can also filter it out your annotation data sets on the basis of whether you can view from the biological process, molecular function and the cellular component for Panther also, and the reactome pathways. So now in this video what we are doing, we are looking for the individual genes, like how we can identify the genes which is related to our database. So basically we start with the Parkinson disease, that is a neurological disorder, or that is a neurodegenerative disease, and by which we have a lot of data is present. So let's start our video and we will see how we can navigate the things. So in this video we just have to find out how we can identify the various research and individual genes on the basis of this. So we are working on the Parkinson disease since. So first we have to, so the first is looking for our research papers for genes list. Okay, so the first is we are looking for the research paper for gene list. So what we have to do is we have to identify the genes related gene list related to Parkinson disease, and we identify the papers. Then we have to go to the particular papers, and what we have to do is, suppose this is the paper where we find the genes, the PARK1, PARK2, confirm linkage analysis, PKR1. So what I have to do is I fetch the genes from there and create a separate Excel. On the basis of that, I will map with our data. Again, I will map with our data and looking for the genes it is present in our data and not. If the genes is present is in our data, and then I will map with them. If the genes is present in our data, so we have to check what is the p-values, either it can be a statistically significant or not, and statistical significant or not. And then we identify the gene. Suppose this is the gene DLG1, and then we will look for the DLG1 supporting a research paper related to Parkinson disease. Okay, the Parkinson disease is available, and this, then I have to find the particular gene which I am looking for. Those genes is not present over there, but still I can go through, because sometimes they give the table in the image forms, or they sometimes it is present in the supplementary. So yeah, this is the one way to create looking for the individual genes which is related for the databases. Start. This is the one way to look for the individual disease for the research purpose. Second is to different databases. So the different databases, like we have to find out the genes information, like is that particular gene is associated with the particular disease. Suppose if we are working on a cancer, so either it can be a work on the cancer or not. So suppose just if we are looking for DLG1 gene cards, or just we can do simply type DLG1 and look how it is associated with the other things. Okay, so it also gives the uniport, OMIM we have to check, and gene card. So we look for the gene card also. So first we're looking the gene in the gene card, and we identified like how the gene is associated with. It's a disk large MAG UK scaffold protein 1 SAF1, and we go for the lower. It is also present in the NCBI gene. We can click over there. Yeah, it is opening. Ensemble ID, OMIM ID, and this summary. The gene of multiscaffold required for normal development. The protein rotated in the function, signal transfer, cell proliferation, synaptogenesis, and the lymphocyte. So what is synaptogenesis? I think this is related to some neurodegenerative disease. Okay, neurons. So synaptogenesis refers to the formation of synapses, that points of contact where the information transmitted within. So synaptogenesis in Parkinson's. In recent years, numerous causative genes risk factor for Parkinson have been identified. More half appear to function of the synapses. Synaptic defects are thought to the prevent neural birth death. So this is the way, like how these genes are associated with the neurodegenerative disease or the particular disease. But you can also go to the direct findings with the help of research paper, that someone is reported that particular gene we are looking for, or the genes which is having a higher expression values is present in the research paper or not. Then we have to move forward. Gene cards for DLG1, autism spectrum disorders, epilepsy. All these things are related to the neurodegenerative disease, because epilepsy is also a neurological disorders. So in the beginning, what we are doing is we are looking for the samples. When we are looking for the samples, there are neurological disease control samples. So definitely if we, instead of control, if we are using our Parkinson disease samples and compare with the neurological disease control, so definitely this particular DLG1 is having a greater higher expression value as compared to the normal control, because it is also a neurological disorder and it is directly reported with the epilepsy. And unblocking NMDA receptors determines MAPK finances. So when you go to the lower down, and you can also the promoters and the enhancer for this particular genes, you can also download the gene enhancer, but the data is a 2017 data. You can also see the genomic bands and which chromosome location it is associated. It is in chromosome number 3, so you can see it is in the chromosome number 3. The reference sake is also giving some attributes, the structures of this particular genes, the identifier. A lot of things is there, so you can come and go through with all the things and see how you can use this tool or this databases according to your research. Okay, now we can move to the Uniprot blast. So we can see again Homo sapiens, keyword, host virus interactions, recommendations, synapses, cellular eukaryote. It is also gives the detailed information. It also gives us the SNP number, so we have to cross check with the SNPs also, like this actually particular SNP is associated with this particular gene, because one genes have many specific SNPs, but yeah, they also give the binary interactions and all. Now we check with OMIM. In OMIM you can see disclose this cloning expression, gene functions, immunoblast analysis of immunoparticipants, focytes, secret actions, additionally tyrokinases. So this is the research paper by which this particular gene function is written in this particular database. It is mapping with the how it is mapping with others, animal models. So this is the way you can find this particular gene is actually associated with your disease or not. So this is it for the today, and yeah, we will see you in the next playlist or in the next video, where we do some end-to-end projects and explaining how we can manipulate the data in detail by using the Python code, by using the Excel sheets, and then looking these individual genes, like the tools or the database which I am using in this particular video or the previous videos. But yeah, we will explain in detail. This is the rough overview in this particular 4 to 5 videos, the GEO videos, but yeah, we will look for more in the upcoming playlist. So definitely see you in the next video.